All right. Now we're getting started. Welcome everybody. I've loaded everything, the questions and whatever we want to do. So let's get to work. Let me go to this page. If you have any question, make sure you stop me and you ask me. I'll be taking all your questions. Okay? So remember, we are doing inheritance part 2, and we'll see some questions together. So let me just quickly summarize the whole thing so that you can all be on the same page as me. So when we do inheritance part 2, there are some things that we need you to know. Okay, this is the second part of inheritance. There are some things that we need you to know, right? The first thing that I try to have everybody know is some important things, some important definitions to say, okay, what is, everybody knows what inheritance is. When you transmit genetic information, when you transmit genes from one generation to another. And then we saw what an allele is. Then we also saw what do you mean when you say genotype. Remember, genotype, we said all the genes and all the alleles that are inside of you. Then we said the phenotype is what we actually see. Okay, what do we see? The observable trait. Then we said homozygous means you have the same. So let me write the words, the important words that I need to make sure. The important words maybe are just like 5 or 6. Okay, the key terms, then we talk about those Punnett squares because they'll be using those. Remember, I need everybody to know these key terms, right? Key terms, and then we look at the Punnett squares. Number 1 is what is genotype? Number 2, what is phenotype? When we are talking about this, we said when we say genotype, we say the genetic makeup, the complete genetic makeup, the complete genetic makeup of an organism. Remember, we'll be looking at one gene at a time, right? We say that the complete genetic makeup of an organism, right? In terms of including all the alleles of the gene, okay? Of the gene which we are looking at. So we look at all the alleles that are there. What are all the alleles that are inside this organism? So when we say genotype, we are talking about the things that are inside of you. Remember, we said that the things that are inside of you, some of them are there, but we can't see them. They are not expressed, but they'll be there. We'll see how do we check. Okay, we're going to see a way of checking. But remember that, everybody, please remember that when we say genotype, we're talking about everything that is inside of you in terms of genes. You can have the gene for black hair and the gene for red hair. And then when we look at you, you may have black hair. But inside of you, having the gene of red hair and the gene of black hair together inside of you. So you having black hair does not mean the only gene inside of you is black hair. It just means that the gene of black hair or the allele of black hair inside of you is dominant. It is the dominant one. But it doesn't mean it's the only one there. So when we say genotype, we want to know every allele that is inside this organism. When we are looking at whatever we are going to be looking at. Then when we say phenotype, now we are talking about the allele that is expressed. Okay, which one is expressed? The allele that is expressed, that is what we say our phenotype, the observable part, what we can see. Are we together? Are we together, class? Hello? Then we said we have the word homozygous. We said when we say you are homozygous, you can either be homozygous dominant or homozygous recessive. Homozygous, it means you have two, it's having, homo means the same, okay? So homozygous means you're having two identical alleles. You have the same alleles, two identical. So your dad and your mom gave you the same allele. Your dad and the mom had the same allele for that gene, whether it's the gene for blood type or it's the gene for hair color, or it's the gene for eye color. Your dad gave you brown eyes, the allele for brown eyes. Your mom gave you the allele for brown eyes. So if you have the same alleles for the gene is the gene for eye color, right? You have the same. You have brown eyes and brown eyes from both your dad and your mom. That means you are homozygous for that particular gene. So having two identical alleles of a particular gene, that's being homozygous. So when you say anything is homozygous, we want to see it having the same alleles. If it doesn't have the same alleles, then I don't know what you're talking about. Because homozygous means the same. When you are homozygous dominant, let's put an example. Homozygous dominant, it means both. Okay, it means the identical alleles are the dominant one. Because you have identical alleles and they're both dominant. IR. Let's just say that. Usually they say XR. We'll see when we talk about sex-linked characteristics. Look at this. Both alleles are the same. You can say, IA. These are both dominant. Then homozygous recessive. I don't want to waste time on this. Homozygous recessive, I'm almost done, means both alleles are the recessive ones. So you'll be having, for example, IO. When you talk about blood type, we'll talk about it just now. Okay? Having the same alleles, having identical alleles that are both the recessive allele. That's what it means. So when you're homozygous, it means you have the same alleles. We would want to know, is those same alleles, are they the dominant ones like here? Are they the dominant ones like here? Or are they the recessive ones? Having the same allele but it's the recessive one. We would want to know that. We'll see. Then anyway, when we now say we have an organism that is heterozygous. Heter means different. So we say heterozygous now. Okay, heterozygous. What does this mean when you are heterozygous? It means having two different alleles of a gene, of a particular gene. It just means the two alleles are different. Just means the two alleles are different. When you're homozygous, the two alleles are identical. Okay? All right. Now, let me say this one. If two identical homozygous organisms or homozygous individuals breed together, if you have two homozygous individuals that are homozygous the very same way, both are homozygous dominant, IA meets another individual, what that is also IA. So IA meets another IA, right? IA crosses with an individual that is also IA. So can you see, this is a homozygous individual, okay? Because he has the two alleles are the same. And he meets another homozygous individual that is also having the very same alleles as that other one. If they breed together, okay, then it will be, if this happens, then it will be pure breeding. So this is what we call pure breeding. You make a pure breed. Are we together? Class? Okay. So now I think all of you are good. Huh? Now I think most of you are good. You understand this. You can even represent your alleles with letters of the alphabet, right? still fine. Make no mistake about that one. So yes. All right. Now, anyway, maybe the last thing I would love to make everybody know is when we say we're dealing with monohybrid inheritance, we'll be just looking at one particular trait only. We won't be looking at more than one trait, right? Now let's start the Punnett squares. So this was background, and I wanted you to then know all the Punnett squares, then we start to do questions. Now let's start the Punnett squares now. I want to do the different parts of the Punnett squares with all of you to make sure you know all the different types of Punnett squares, right? I'm going to do with you 4 types of Punnett squares and the pedigree chart, then you'll be done. So let's say here the 4 Punnett squares. The 4 sections of Punnett squares or genetic diagrams, so that you understand when we get a question in the exam. Write with me. The 4 sections of Punnett squares or genetic diagrams. So what do you need a genetic diagram for? You want to predict the offspring that is going to be made. That is all we are trying to do. We want to predict the traits that are in an offspring. So that's all we want to do. We want to predict the traits that are in an offspring. Okay? So the first section which we're going to be doing, let's put here the first section that we're going to be doing. Say the basic monohybrid genetic crossing. Monohybrid genetic crossing. We are just doing the basic monohybrid genetic crossing, okay? We're just crossing basic things. Let's do it. The first one which we need to do is, let's just say you're going to have let's put the situation that we have here. First, I want to put the situation, because in the exam you're going to be given the situation, right? Then when you give the situation, they'll tell you, hey, we'll see the questions that they come. But first, they'll tell you a situation. So let's say B is going to be representing, we are analyzing let's say we are analyzing what? We're analyzing the gene for eye color. So the gene for eye color has the following alleles. I'm going to write the situation, then we do the 3 tables you must know. Has the following alleles, or we can even do 4. Number 1, B is dominant one, and it is for brown eyes. So B is for brown eyes, and it is the dominant one. That is why it is in capital. Then small b is the allele for blue eyes. So small b is for blue eyes, and it is recessive. So this is what we have, okay? This is the situation that we have. Now let's start to do the crossing. So have you seen the alleles for eye color? Have you seen them? Big B is what? Brown eyes, the dominant allele. Are we together? Hello? All right. Then small b is the allele for blue eyes, right? Small b is the allele for blue eyes. Now, what we do know is that when we say we have an allele that is dominant, it means if it is present, it is going to be expressed. Okay, an allele that is dominant, if present, it is always expressed. So for you to have a recessive allele expressed, it must be the only one there. If dominant is there, it's the one that is expressed, so we'll see it in the phenotype. Anyway, now let's put the first situation now. Situation number 1. We want to be doing the genetic cross, okay? So the first one we're going to do genetic cross. They'll tell you the genotype they want. We want to do the genetic cross between 2 homozygous individuals. 2 homozygous individuals. One is homozygous dominant. Then the other is homozygous. The other is homozygous recessive. is homozygous recessive. So you say, what are you talking about? What are we even doing? Right, so this is the second section of inheritance. The only thing you must be able to do, hey, listen, you can do this. The only thing you must be able to do for this second part of inheritance is you must be able to check what the probabilities, okay, it's like the percentages. We'll talk about that one. That is easy. But you must be able to predict the type of offspring that you get. You must be able to predict the type of offspring that you get. Yes. Yes. Roderick? Yes, I want you to master this. When we say the dominant one is expressed, right, we mean that when you look at the child, what you see is the phenotype. Listen, there is genotype and phenotype. So we are going to see what is in this child in terms of genotype. Then we are going to see what will human beings see with their eyes. So what you must know right now is this. If I see you having brown eyes, it doesn't necessarily mean you don't have a gene for blue eyes. But maybe the brown eyes you have will be the dominant allele, so you end up having brown eyes. Do you understand? So what you always see is the dominant one, if it is there. So if you have a person who has red hair and black hair inside of them, and red hair is the dominant one, when you look at their hair, it's red. Because inside of them, they had red and black, but red was dominant. So it is the one that is expressed. If they had red and black is the dominant one, you will see the child having black hair. And yet that child has a gene for red hair and a gene for black hair. All right, so anyway, let's see. They're going to give us the situation. We're going to be practicing with situations, then all of you will get a lot of experience. So let me move fast, okay, because I want to do those examples so badly. So here they'll tell you, first of all, they will tell you the situation you have. So they are telling us, hey, here is what you have. This is the situation you have right now. We are dealing with the gene for eye color. And the alleles there are these ones. So they will tell you. Then now we can come here and they can now tell us, find the cross between this and this. Right. Now let's do the cross. Okay. We'll study the types of questions they give us. Don't worry. So here you should draw your table very fast. You do it like this, eh? Then like this. Like this. Right. It's open like this. So now what you're going to do here is you're going to say, this side we have parental genotype. You can put the parental genotype this side. They say one is homozygous dominant. So if it's homozygous dominant, you take the dominant one, which is B, capital B. So you say this person has B and big B, two big B's. Right? This person has two. Can we see that? Hello. Then now what we're going to say is we have this another parent here. So you just write parental genotype, which means the things that are inside the parent. Here they say this other parent is homozygous recessive. Homozygous means the same. Recessive means they have small alleles, the recessive alleles, which is these ones, small b and small b. So these are the alleles in. Remember, you always have two. Each individual has two. So you should know, are they heterozygous? If they had said we have heterozygous parent, it would be like this. Big B and small b. That's what it will always be. And you always start with the big B. But here they said homozygous dominant and homozygous recessive. So now we will put here the gametes. We'll put the gametes. In here it's big B and big B. In here it's small b and small b. Then we start crossing to see what type of children are we forming. Here we have big B meeting small b, so you have big B, small b. Then here we have big B meeting small b, you have big B, small b. Then here we have big B meeting small b, we'll have big B, small b. Then here we'll have this big B meeting this small b, we'll have big B, small b. Now, what is the genotype of the offspring? The genotype of the offspring. In other words, they're asking you, what results do you get for the offspring? You can see that all the offspring will be big B, small b. That's the only genotype. Then what phenotype do you get? What phenotype is this? In other words, if a person has these genes in them, what type of eyes do they have? Because the dominant one is there, it's the one that will be expressed. So this person will have brown eyes. This person has brown eyes. And what should you say about the probability? What do you say about the probability? You can say now that 100% of the offspring, all the offspring, 100% of the offspring are heterozygous. 100% of the offspring will be heterozygous. If these are the parents, if the parents are homozygous dominant and homozygous recessive, the children, all the children that are made from such parents, all the children are heterozygous. Are we together? All the children are heterozygous. All right. Heterozygous just means the two alleles are different. Are we together? Hello? So should we go to the next one? Hello? Okay. So now let's do the second example. We are still in part 1. Hello? We are still doing the basic ones. We're going to do the other ones that I want you to know. We're still doing the basic ones. The part 2 now is genetic cross between, they'll tell you now, please give us a genetic cross, okay, between two individuals with heterozygous genotype. We now want the genetic cross between two individuals that have heterozygous genotype. So what do? Make your table. You say 1, you say 2, you say 3 lines. Then here you come, you say 1 line, okay? You say 1 line like this. You say 2 lines like this. Then you put the third one closing nicely like this. Then in the corner here, that's where we have our gametes, okay? You can leave it blank if you want. Then here you say now parental genotype. Parental genotype for the first parent, both of them are heterozygous. So it's big B, small b. Then here again, parental genotype. Parental genotype. Here you have big B, small b. Are we together? So now let's separate the gametes. We have big B and small b. So we put the gametes big B, small b. Then here we have again big B and small b. We put the gametes big B, small b. Now we start the crossing. Big B meets big B in here. You have big B, big B. Big B meets small b. You have big B, small b. Then in here, big B meets small b. You have big B, small b. Then small b meets small b at the corner. You have small b, small b. Can you see this? Can you see this? Here we have, this is the genotype of the offspring that we are now getting in here. So now if they then ask you, please tell us the genotype of the offspring that you get. We got big B, big B. We got the genotype is going to be big B, big B. We got big B, small b. And we got small b, small b. What about the phenotype? What is the phenotype that we got? Here, big B is brown eyes. Okay, big B, small b again is brown eyes. Why? Because the dominant one is there. Then small b is blue eyes. There's another one for blue eyes as well. All right, so now what do you do here? Can you all see? Okay, I'm happy. So now we're going to say, what about the probability? You can say there is how many do we have of big B, big B? It's 1/4. So there is a 25% chance that the individual will be big B, small b. Okay? Then again, there is what about big B? No, this is for big B, big B. There is 1/4 chance. And there's also a 25% chance of having small b, small b. And you also have now the 50% chance there's a 50% chance of getting a big B, small b, because there are 2/4, which is half, which is a 50% chance. Are we together? Are you seeing this, students? Are you seeing this? We are using the situation we have been given to create these genetics. We are using the situation we have been given to use this to make these Punnett squares. Let's make the last one here, which you have been given. They want the last one. Say genetic cross between an individual with a heterozygous genetic cross between an individual with a heterozygous genotype and a homozygous recessive genotype. This is so easy. It's almost like you say, I don't want to do it. I know what's going on. Come on, can't you make it harder? So we'll say 1, 2, 3. Then we'll say 1, then 2, then 3. Like this. And here is like where you have the gametes or whatever. Then this is parental genotype. Okay, we have one that is heterozygous, so it'll be big B, small b. Then we have again here parental genotype. Okay, here is small b, small b. Now let's put the gametes. It will be big B, small b. Here it will be small b, small b. Can you see this? We now say big B times small b, you get big B, small b. Big B times small b, you get big B, small b. Small b times small b, you get small b, small b. Small b times small b, you get small b, small b. Okay? Do you understand now? Do you understand? Yes, you can understand. They can ask you to do between heterozygous and one which is going to be homozygous dominant. That doesn't matter. You can still do it. And then if they ask you for the genotype that will be present, the types of genotype, you can see that we have big B, small b, and we have small b, small b. Then if we see the phenotype, here, phenotype big B, small b, this is brown eyes. Can you see? Brown eyes, and then we also have small b, small b. This is blue eyes. Well done. Yes, you're doing correct. Well done. So here we are done with this first part. Can everybody agree with me that the first types of genetic diagrams we are fine? Can you all agree with me, class, as a class? Come on now. Hello? We are fine with this, right? We can get, yes, there's a 50% chance that we'll get brown eyes. So you can already see that there's a 50% chance that you get brown eyes. And there's also 2 blue-eyed people, so there's a 50% chance that you get blue eyes as well. This is what happens. Are we together? Now, let's go to the second type of thing. Second section of genetic diagrams, we have what we call a test cross. Can everybody write this? This is the second type of genetic diagram which you must know. We're going to do 4 of them. We've done the first ones, which are the basic ones, where they'll give you the gene and the alleles that are present, and then they'll ask you to construct genetic diagrams for a particular situation. Now, what about test cross? We are now in section number 2. We have 4 types of genetic diagram sections that we want to cover. We are done with section 1. Section 2 is now about learning the second type, which is a test cross. What do we use it for? Write for me here. This is used to know genotype. This is used to identify unknown genotype. Used to identify unknown genotype. You want to know, is it homozygous? Okay? Is it homozygous or heterozygous? Do you get it? Right. Is it homozygous or is it heterozygous? You see, let's say you're looking at an individual, and this individual that you're looking at has brown eyes, but you don't know this brown, is it big B, or is it big B, small b? So we want to be able to find out the genotype. How do we find that? That's big, right? So here is how to do it. How to do a test cross. There are many ways, but here is one. Cross with a pure breed. You cross with a pure breed recessive genotype. Are we together? With a pure breed recessive genotype. Okay? You are going to cross with a pure breed recessive genotype to try and find what is missing. Do you want to see an example? Guys, we want to do a test cross. We're looking at an example here. So we want to find out. Let's say you want to know, class, you want to know what is the genotype. What is the genotype? So what I'm going to do, I'm going to show you the 2 types of tables that are there. Look, you have a person who has brown eyes, but you want to know, is he homozygous dominant or is he heterozygous? You bring the recessive arrow and you cross. So let's see their genetic diagrams, because you need to know. Let's see the genetic diagrams of the 2 situations. We're going to split it like this so that you can be able to see the genetic. So here I want you to say parental genotype. Don't use up space. Parental genotype, let's say we have big B. Okay? We are going to cross it. We are going to cross it no matter what. So now what we are going to do, here is the other parental genotype. What did I say is the other parental genotype? When we are doing a cross diagram, when we want to know what is the type of thing, when we do a cross diagram, or a test cross rather, sorry, we want to identify an unknown genotype. We cross it with homozygous recessive. Let's see what we get here. Okay? Let's see what we get. If we cross with big B, big B times small b is big B, small b. Big B times small b is big B, small b. Big B times, this is going to be big B, small b. Okay, it's B, small b. Can you see? For here, it is this big B meeting the small here, big B meeting the small thing. Here, big B meeting the small b. Here, big B meeting the small b. Can you all see that? So you can see that 100% of offspring, if you are homozygous dominant, when we put on a test cross, we should get 100% of offspring being heterozygotes. Okay? Okay? Can you see? Anyone with another question? What if we cross a het? Let's say the parent was not homozygous. Because we don't know, we are going to look at the type of offspring that is got, and then we'll be able to predict. Are we together? So now we are going to now check what type of offspring do you get when you have a parental genotype of a heterozygous. So here it will be like this. Then here, then you are going to have again parental genotype. Okay, parental genotype. Let's go. Here, of course, is the homogeneous. There we go. Homogeneous recessive. Here we want heterogeneous. So here we'll say, what are the things that come in here? Big B, small b. What about in here? Small b and small b. Now we start crossing. Big B meets small b, we get big B, small b. Big B meets small b again, we get big B, small b. Small b here will meet the small b at the bottom, it will be small b, small b. Small b there will meet the small b at the bottom, it will be small b, small b. So in here now, if you get 50% being heterozygous, then you are going to have 50% in homo. So here you get 50% homogeneous recessive. Okay? Can you see this? 50% will be homogeneous recessive. Have you seen this? So analyze this test cross. You must know it. Okay? If we don't know the genotype, we're going to do this, then we'll be able to know, right? We've seen that if you have a heterozygous individual, you cross him with homogeneous recessive, you get 50-50. Okay? Can we move on now? Hello? Can we move on to the next thing? So here we saw 100% of offspring are heterozygous or have brown eyes. So here we have it, okay? This brown eyes 50%, then the other 50% blue eyes. So here 50% blue, 50% homozygous. What did I write? Homogeneous? Okay, so this is 50% will be homozygous recessive, that's blue eyes. Homogeneous, we said it yesterday, it's for solutions in chem. Don't ever let me write that. That's never supposed to be right. The case. Are we together so far? So have you seen the second part of Punnett squares? It's on test cross. Have you seen that? All right. Have you seen that? Now let's do the third part. It's going to be on something called codominance. Codominance. This is self-explanatory, right? It means we have two dominant alleles, but there will be different alleles. So they're both dominant, yes. So the mom gave you a dominant version, yes. And the dad gave you a dominant version, yes. But they're actually different versions, yes. Whew. Normally we know if the mom gives you the dominant, the dad gives you the dominant, is the same. There'll be homogeneous, there'll be homozygous dominant. Now, how can we have this? Well, it's a special situation. So what is codominance? And then we'll see a diagram we think about. We've seen for test diagrams, the most important thing is that you cross with homozygous recessive genotype. Are we together? And you see the results. Why are some of you quiet? Is there a problem, class? Hello? Why are you quiet, people? Are you all fine? What I'm saying, does it make sense? Like, are you understanding? Are you understanding what I'm saying? All right. Now, let's see the Punnett squares for codominance, because they're going to give you one of these in the exam. So what is codominance? They can even ask you to define it. Codominance is we are dominating together. So it's like this. You see it sometimes in chickens. The father is a black one, the mom is a brown one. Then they give a child that is black and brown. So that means they're sharing. Usually it's not like, it's like you get let's say you have red and you have white, you get pink. That means both are expressed at the same time. So they share. They both express at the same time. That will be codominance. So codominance, right, to say it is a situation in which both alleles, it's a situation in which both alleles in heterozygous organisms, hello, contribute or are expressed, you can say are expressed, or contribute to the phenotype. Phenotype is what we see. So, when we have a black and white person, we get somebody who's in between. It's like both are expressed at the same time, something like that. Okay, so both are expressed at the same time, yes. So that's codominance, yes. What situation should you know for IGCSE that is an example of codominance? It's blood groups. So let's say, okay, let's put IGCSE example. Here is the example you do on codominance. It's the inheritance of blood groups. Inheritance of blood group. If you get a question on this, they are asking you about codominance. Okay? Are you hearing me? All right. So let's talk about the situation. You don't just cross. You need to know what is the gene involved. So here we are dealing with the gene of blood groups. What are the different types of alleles that are there? We know that if we're talking about hair color, we would say, oh, we have the allele for red hair, we have the allele for black hair, we have the allele. So what are the alleles we have for blood, for the blood groups? Okay, there are 3 alleles now. Here's the situation we're already giving you. There are 3 alleles. And the alleles are IA, they are expressed like this. IA, IB, and IO. Now, what we know is that even if you go to China, or if you go to Japan, or if you go to Jamaica, or if you go to Mars, no, there's no one on Mars, but if you go anywhere and you meet a human being, you won't meet a human being with 1 allele. Okay? So these alleles, a human being will have 2. Are we together? Class, are we together? So a human being will have 2. So let's see now what these are, which one is dominant and which one is recessive, so that we will know. Because when you are given a situation, you should be told the alleles that are there, and you should be told which one is dominant, so that you know, you can predict the phenotype, isn't? So let's put here, there are 3 alleles. IA, this allele, okay, blood group A, and blood group B, okay, and blood group B are co-dominant, which means they are both dominant, okay? So when they come together, they will be co-dominant. What does it mean? Both show their effects. Both show their effects when present together. Okay? When present together. So when present together, both will show their effect. Are we together? This is the first time you are hearing this. So hey, I know you haven't heard about codominance, but we've already talked about it. So just know that when we talk about the phenotype here, both A and B will be the phenotype. Oh, really? Yes. Normally the phenotype is the dominant allele, right? Yes. But here, both will be together. All right. Now let's go now to the second one. Okay? IA and IB are both dominant over the IO allele. So if IA is present with IO, you see A. If IB, which is blood group B, is present with blood group O, you see blood group B. That person will have blood group B. Although inside them, in their gene, they had instructions for both B and O, but O is recessive. So the only time you see O is when it's just there alone. So now let's put the different types of genotypes we can get. Okay? Let's put the genotypes that we can get. You can have IA, or you can have IA, IO as your genotype. For both of these, what is the phenotype for here? The phenotype is A. So this person will have blood group A if they have these two genes. What? What are you talking about? Since when do we just guess? We are not guessing. We have, when we have the blood group A alone inside of you, let's say you just have, that is so important. That is why people actually specify, can I have this blood group type? You have to say, okay? So if you have these two, hello? If you have these two alleles, IA and IA, which is blood group A and blood group A, only, then your phenotype will be blood group A. That means you have blood group A, Mr. Man. Those are the genes in you. So what we will see in you is blood group A. If you have the gene, or if you have the allele for blood group A and the allele for blood group O, remember we said A and B are dominant when they meet this one. So this one will not be expressed. We will just see blood group A. If you have blood group B, the allele for B, and another for B, or if you have the allele for blood group B and blood group O, then what we see is blood group B. This is what we will see. This is what you will be. Are we together? See. So where is codominance here? You haven't talked about codominance. Are you not supposed to be preparing us for a Punnett square where we'll meet these type of alleles and we meet A and B crossing and we are supposed to see codominance? That's what I'm about to show you. What happens when blood group A meets blood group B? What is the phenotype? Phenotype is AB. They will be expressed together. This is the codominance. Are we together? When A meets B, they will be expressed. So that person will have the phenotype AB. So that will be their blood group. Now, lastly, the last type of individual is an individual that has blood group O and another allele for blood group O. That person in their phenotype will be blood group O. So that person will have blood group O. Are we together? Now let's do the last type of Punnett square. Hello? You're not going to let me down. Only 4. Hey, you. The last type of Punnett square is when we do sex linkage. Right? Sex linkage. We want to know the Punnett square for sex linkage. They can ask you. Or sex linked characteristic. Sex linked characteristics. Want to see a sex linked characteristic. In other words, something which is going to be appearing in one gender. Either we want to see something which is going to be, yes, yes, it doesn't appear on everything, yes. Yes. And they are different. 2 different dominant alleles coming together. And for IG, we'll only do the one for blood groups. So when we meet this, we will never be shocked. We won't be shocked, right? All right. Now let's talk about sex linkage. Then we'll see the one which we'll meet, which we expect to meet. Sex linkage. Write for me this. A sex-linked characteristic. We said codominance, it's when both alleles are expressed in the phenotype. Both alleles are expressed in the phenotype. A sex-linked characteristic. What is this one now, huh? Mr. Clarify. What is a sex-linked characteristic? When we say we have a sex-linked characteristic, okay? Sex-linked characteristic is a feature in which the allele responsible, can say the gene responsible, is located on a sex chromosome. Are we together? Or we can say the alleles responsible are located on a sex chromosome. So either they will be on the X chromosome or on the Y chromosome. Are we together? Hello? So you'll be having something, if you have anything like that, any condition that is located, any condition that has its allele, the allele that brings it, that codes for it, the instructions, the allele has the instructions for that thing. So the allele that codes for it, if it is located on a chromosome, then you must know that that thing is sex-linked. It will appear in one of the genders. So if we have something that is appearing in one of the genders, that's a sex-linked characteristic. Are we together? So we'll be now saying that the other gender will be just a carrier, but it will only affect the other gender. If it affects males, then the females are carriers. They are not going to be affected, but the males are affected. Are we together? Are we together? So, yes. Men, yes. Colorblind. Men are colorblind. Yes. Men are colorblind, not women. So the reason why men are colorblind is that this allele for colorblindness is on an X chromosome. That's the one which you do for IG actually. So let's go ahead and let's see it. So let's go. There are some men that are colorblind, but never a woman. Let's go here. So responsible, it's located on a sex chromosome. This makes the characteristic. This makes the characteristic more common in one sex than the other. That's just what sex-linked characteristic means. It means it will be a characteristic that has an allele that is located on a chromosome. Okay? On a sex chromosome, not just a chromosome, but on a sex chromosome. All right. So let's do the IGCSE example. Here's the IGCSE example for this. For codominance, we saw it was blood group, the blood groups. Right. Let's now see here, and let's see what are the alleles. Let's see the condition. Then we see the alleles for that condition. Are we together? We want to see the condition, then we want to see the alleles that are there for that condition. Are we together? Hello? All right, good. So let's put it. So let's put it now. It's going to be, all right, just this example. Red, green, color blindness. Okay? A red, green, color blindness. So I want you to say, this is an example of a sex-linked characteristic. It appears in one gender more than the other. Are you together? So let's say this. This is a genetic condition caused by mutation of the genes located on an X chromosome. So because they are on an X chromosome, they affect males and not women. All right, so example. No, so he say here, the alleles for this condition are, you have XR, which is, okay, let's put it, XR, this is a normal allele, and it's the dominant one. Are we together? The normal allele is the dominant one, okay? Then you have this XR, which is the color blind allele. The only time it's seen, it is the recessive one. The only time it's seen is if the dominant one is not there. Are we together? So now let's put parental genotype. So we're going to say consider a cross between a normal father and a carrier mother. Okay, or normal male. Right, let's just say normal father and carrier mother. It still doesn't matter. So let's put it. What will be the genotype? Let's put carrier mother. If the mother is a carrier, then what does she have in her genotype? What is the genotype of the mom? That means she has XR and XR. She is also normal, but she's just a carrier, right? This woman is not colorblind. Why is she not colorblind? Because the dominant one is there. Can you see that? Right. So this person is going. Now let's say normal father. Let's put the normal father here. If the father is normal, remember men have X chromosomes and Y chromosomes, right? So the normal father should have the normal for the X and the Y. This is what a normal father will be, and that's what a carrier mother will be. Are you seeing? Because you can be asked to make something like this. Have you seen how it is like? The normal father, for him to be normal, he mustn't have this one. If he has this one, then he's going to be colorblind. So he should have, remember, men have X and Y. Women have X and X chromosomes. Right, now we're going to put their gametes. So it's going to be XR from mom and another XR. Then from the dad, it's going to be XR, then a Y. So let's see what we get here. We cross, we're going to get XR, XR. What is this class? What gender is this person? XR, XR. What gender is this person? With two X chromosomes. It's a woman. It's female. And is she colorblind? No, she's normal. Okay. What about if XR meets this small XR? It will be like this, then like this. What type of what gender is this person? She's female. And she's a woman. And is she colorblind? No. Let's see here. When Y meets this XR, you're going to have XR, then Y. What gender is this thing? Male. And he's normal, right? Right. Now, let's see when Y meets this one now. We'll get XR and Y. What is this? This is a colorblind male. Can you see that? This is now a colorblind male. Can you see that? Hello? So this is a colorblind male. Yes. So you can say there is a 25% chance. They ask for probability, okay? That the offspring is a color blind male. Yes. Yes. So you can say whenever you have 4 kids, you expect one of them to be colorblind. They'll be colorblind, yes. They'll be colorblind. They'll have this red-green color blindness. Questions? Questions so far on things you've seen? Do you have any questions, class? Huh? Oh, no questions? Really? All right, now we are done with the genetic diagrams, okay? We are done. We now need to just answer questions and use some reasoning. Let me just say something like this. Let me just say something to all of you. When you are now going to meet something called a pedigree chart, let's now talk about this. It's not a genetic diagram. Let's talk about a pedigree chart. This is the last thing I need all of you to know. Can everybody have pedigree chart, please? Pedigree chart. You want now to talk about a pedigree chart. Okay? You want to talk about pedigree charts. I want to talk about the pedigree chart. Pedigree chart, can you see? What is this? Say, I want you to just write this, to say, this is used to track to track a trait. You want to track how is a trait passing through generations. So you want to track how a specific trait or disease or condition rather, or genetic condition is passed through generations. Okay? Is passed through generations in a family. Is passed through generations in a family. So I'm going to say, all right, can I have grandfather? Grandfather, can you come here? Grandfather will stand. I'll make him stand with grandmother. Then I say, okay, show me the kids. Show me your kids. Then their kids will stand in line. Then I say, okay, so for each of these kids, please show me who you married and your kids. So these kids, they'll take who they married and they'll show you their kids. This one takes who they married, they'll show you their kids. This one they'll show you their kids. Now you start following. Okay, in this whole family tree, who has asthma? Then they raise. You check like this, you check like this. Then you make a pedigree chart. Are we together? Hello? Are we together? So the pedigree chart will be showing, you'll be tracking how a specific trait or a disease has passed in the family. Okay? So why is this important? Why would you do that? Because this can help predict the chances. This can help predict the chances of inheriting, of you inheriting the genetic disorder. Okay? The genetic disorder. Are we together? Hello? Are we together? Yes. Yes. Will be what? Yes, if the pedigree chart is showing that, then you need to work carefully, okay? You work against it to say, okay, so this person in this family, in my family, this person and this person and this person and this person had this condition. So every time I need to continuously go for testing to make sure. I don't want it to surprise me. I want to always stay ahead of it. So that's kind of what it is, okay? Because some of these things, you can't run away from them. I wish I was a doctor. This is as far as I know. Roderick, do you want me to lie to you? I don't know how to reduce the chances. Okay, Roderick? You can now research. Roderick, are we together? Okay, good. I don't want to lie to anyone. And then you come and say, sir, you told me this, but that's not true. Maybe when you're a doctor, you'll be like, when I was 16 years old, our teacher once told us a lie. I don't want to do this. Okay, so let's just talk about a pedigree chart. Let me say this. Here's what you see. The box will be representing males. They'll actually give you a key, so don't worry. And circles represent females. Okay? And when a box is shaded, or when a circle is shaded, this will be an affected person. So this is an affected male, and this will be an affected female. Do you understand? The person will be having that, whatever the condition. Are we together? Now, one last thing you should just know about a pedigree chart. We'll see how to answer the questions, but you should know that a horizontal line, if you have a pedigree chart, just look at the time. Ish, I don't like time. Time is an enemy, I'm telling you. There's no way this is 10 o'clock. How? How is it 10 o'clock? Anyway, horizontal line between male and female. If you have a horizontal line between male and female, this shows that they had a kid. It shows that they had offspring. This is so important. The moment you see a horizontal line like this, connecting male and female, just know that these two had offspring, okay? Are we together? Once you see a horizontal line connecting a male and a female, that means, hey, doc, you want to show us that these two had offspring? Okay. Are we together, class? Why are you quiet? Are you tired already? Come on. We didn't even go for long. Come on, guys. Okay. Right. Now, let me say one last statement that can help you. That is a very important thing. Okay? If two parents without a condition, if two parents without a condition have offspring, if two parents without a condition have offspring with a condition, obviously those two are what? With a condition. Those two parents are heterozygous. Yeah, they're carriers, which means they were like heterozygous, like AA, like something like this. Yeah? These are the parents. So they were not having it, then they have a child which is like this. So this is going to be the child. But it also what it shows is this with a condition. If they have a child with a condition, then you should know that then this is evidence. If they ask you, what evidence do you have that this particular condition is caused by a recessive allele? To say this is not a dangerous condition as long as you don't bring two parents with this thing. Okay? That is why we say you don't marry your relative, because you bring recessive alleles together. But anyway, we are saying then this is evidence that the condition, if parents didn't have, but the child has, this is evidence that this condition is caused by a recessive allele. Because the only way parents didn't have, but the child has, is the parents were carriers, like what Bradley's saying. They had the recessive allele, and the recessive allele from papa met the recessive allele from mom, and it expressed in the child. So this is evidence that the condition, if they ask you to show evidence that the condition is caused by a recessive allele, here is the evidence, okay? Are we together? By a recessive allele. Are we together, class? Okay. Are we together, Mr. Roderick? All right. Are we together, Karabo? Tadisa? It's been a long time, Tadisa. Are we together? Is Tadisa in class today? Antonisha, are we together? Good. Kuzi? Good. Good. Is Inzwi in class? Inzwi, are we together? Ooh, where is she? Okay, anyway, I'm proud of you students. You're very good. As you can see, time is an enemy of progress, and it's robbing us continuously. I was very, very heartbroken when I couldn't go live, but it's fine. I'll see. I'll make a plan to either go live during the night, like deep in the night after my A-level lesson, or to meet you earlier tomorrow. I don't know, because I want to do the things that I wanted to do. I wanted to talk about esters. I've gathered the questions together. I want to tell you about esters and answer questions. And I want to finish the electromagnetic spectrum for physics and talk about dispersion of light and dispersion of light and do questions. And I want to go to chapter 6 for physics. Okay, I really, really want to teach you chapter 6 and also chapter 5, nuclear physics, and chapter 6, space physics. I really want to teach you before you guys get a lot busier. So we'll see how it goes this week. We'll make time. If we have to be together for a long time on Saturday, it's fine. So it's not the time to think about breaks right now, right? You're not getting tired, right? Good. So keep these notes. We're going to do diagrams tomorrow. If you need to rewatch the video, I'll send you the link